Hello everyone. This is Dr. Prarthana, Assistant Professor in the Department of Public Health Dentistry from Vinayaga Mission Sankaracharya Dental College. Today we will have a small insight on the role of diet in dental caries. Epidemiology is a big chapter. Welcome to our presentation on epidemiology of dental caries. Today we will delve into our discussion focusing specifically on critical role of diet, initiation and progression of dental caries. So today we will go fundamentally on both preventive and effective management of it. We will more focus on the diet related programs which happen in dental caries epidemiology. So role of diet has two key components, which is the diet and nutrition. So first, the diet refers to type and amount of food which is consumed daily by an individual. So it's an amount and not the processed individual. This encompasses the entire spectrum of individual eating habit, including meal frequency, portion size and variety of foods. Nutrition is a sum of process by which an individual takes in and utilizes the food. So it is a process of ingestion, digestion and absorption and metabolism and excretion which highlight the biological process of food for energy and growth purpose. So distinguishing between the diet and nutrition is very crucial for comprehending their impact on oral health. So while diet refers to consumption pattern, nutrition describes the physiological utilization of these consumed food. So both aspects significantly influence the oral microbiome and host susceptibility to the caries. So before understanding the diet role in dental caries, we have to understand the role of sugar in dental caries. We have two forms of sugar, which is the added sugar and the free sugar. The added sugar, which are intentionally incorporated into the foods and beverages during their processing. So this category includes common sweeteners like honey, fruit juices, brown sugars, high fructose sugars, corn syrups and many others, and also our day-to-day sugars. While free sugars are the monosaccharide and disaccharides that are added to the food by the manufacturers, cook or consumer, plus sugar which is naturally present like honey, syrups, fruit juices, etc. And they are in the form of glucose, fructose, lactose and sugars in fruit juices even. So this distinction between the added and free sugar is very vital on both contributing significantly to the cariogenic potential of the diet by providing readily available substrate for acid producing bacteria. Limiting the intake of these sugar is a cornerstone for the caries prevention. So coming to the sugar, the amount only doesn't have a direct role. Always there are other factors of sugar like consumption and frequency plays a major role in dental caries. So first, sugar intake. As I said, the frequency of consumption of fermentable carbohydrate plays a major role in direct link of dental caries. Then the bacterial metabolism. As we already know the keys triad or tetrad, there is bacterial metabolism which plays a key role in the dental caries formation. And acid production, the decreased as a pH of the plaque and leading to the demineralization of the enamel, and enamel demineralization, loss of mineral content from the tooth structure, and there is a progressive caries formation. So the direct relationship between sugar and dental caries is already well established. So there are some WHO recommendation for this sugar intake. Initially the sugar was only needed for the energy purpose. Free sugar should constitute less than 10% of total energy intake, and further reduction below 5% is suggested for additional health benefits. Annual free sugar consumption in the presence of fluoride, annual free sugar should be limited to 15 to 20 kg of per person, and frequency, always we have to reduce the frequency and limit it during the meal time. So frequency of consuming sugary foods and beverage should be limited maximum for 4 times per day, and it is recommended target duration of acid attack on the tooth enamel. Vitamins and minerals in dental caries prevention. There are some essential micronutrients and macronutrients which is very essential for preventing this dental caries. First, the vitamin A, which is crucial for development and maintenance of epithelial tissue, including in the oral cavity, supports the salivary gland functions. Vitamin D, which is essential for calcium, phosphorus absorption, and a critical role for tooth mineralization and bone benefits. And vitamin K, a major role in bone metabolism, mineralization, contributing to the structural integrity of the teeth. And vitamin B complex for overall oral health tissue and metabolism, indirectly supporting the resistance to the oral infections. And calcium, phosphorus are a primary structural component of teeth, and their adequate intake, particularly during the tooth development, is a paramount important, and it forms a caries resistant enamel and dentin. And fluoride, as we already know, it is for powerful anti-cariogenic property. While it is associated with water fluoridation and dental product, fluoride is also naturally present in trace amount in leaves and skins of various plants and fruits, contributing to the topical remineralization. Beyond sugar control, a balanced diet rich in specific vitamins and minerals, fortifies the teeth against the caries. These micronutrients plays a vital role in enamel formation, remineralization and supporting the oral environment defense mechanism. And coming to the evidence for direct diet caries relationship, there are various studies which is the main focus of today's topic. The human observational studies and experimental or interventional studies has been formed a direct relationship of sugar or diet intake with the caries. So human observational studies are longitudinal studies tracking population dietary habits and caries prevalence, which will reveal a strong correlation. And interventional studies, there are some specific control trial which has been done for dietary factors manipulating and demonstrating causal link between diet modifications and caries outcome. And there are also other established studies which has been done like animal experiments, block pH studies, enamel lab experiments and incubation studies. Human observational studies, mainly we have Hopwood studies and hereditary fructose intolerance. Both of these are observational studies where a specific naturally observed sugar consuming community has been observed over a period of time, and they have been noted a direct relationship between the sugar and the dental caries and diet and dental caries. And human interventional studies, they are pivotal role in establishing a causal link between the diet and dental caries, and they are controlled experiments which is manipulating the dietary factors and meticulously observe the resulting changes in the caries incidence. So first we have Whipple-Holm study. It demonstrates the frequency and stickiness of sugar consumption were critical for the caries development, so that the total amount of sugar consumed, which is nothing but first the Whipple-Holm study was given by Gutsefer et al. In 1954, and it was a 5-year investigation which was done for the purpose to find if the caries increases with the intake of sugar or the caries decreases with the decrease of intake of sugar. So method used was 7 group of people have been formed, in which 1 is control group and 6 are the experimental group. And here the control group, they had 60 males above 34 years, and it is a 2 years experiment where they received low carbohydrate, high fat diet, and sucrose group had 57 males. They received 300 grams of sugar given in the solution form. And in a bread group, they had third group, there 41 males and 42 females received once daily with the afternoon coffee, 345 grams of sugar bread containing 50 grams of sugar daily during first 2 years. And chocolate group had 47 males who received 300 grams of sucrose, and a caramel group where 2-year controlled group of 62 males, 22 caramels daily in a 2 portions was given. And there is an 8 toffee group where 44 males are given a low carbohydrate, high fat diet and 8 toffee per day. And 24 toffee group where 48 males received 24 toffees between the meals during the 3rd and 4th year. So this study not only gave the consumption of sugar and followed up, but also the frequency and intake of time were also noted. So the main conclusion of this Whipple-Holm study was an increased carbohydrate definitely increases the caries activity. The risk of caries is greater if the sugar is consumed in the form that is retained on the surface like sticky form, and the risk of sugar increases caries activity at the greatest if the sugar is consumed between the meals. Upon the withdrawal of the sugar, the increased caries activity rapidly disappeared, and caries lesion may continue to appear despite the avoidance of refined sugar and maximum restriction of natural sugars and dietary carbohydrates. The increase in clearance time of sugar increase the caries activity. This study showed the physical form of carbohydrate is much more important in karyogenicity than the total amount of sugar intake. So the drawback is the groups were made up from the patients. It is not the common public, the patients were involved, and with no possibility of matching of age. There were diverse age and diverse group which was involved in all the 7 groups, and the patients were mentally challenged also, so they did not always understand or follow the instruction. The dietary regimens of various groups were not changed in consistent pattern. Some group had different and some group have a longer period of change. So this was not giving a proper result with this drawbacks. And the second study was Hopwood study. Hopwood study is also an observational part, but the strict diet regimen in the residential institute provide a strong evidence for protecting effect of low sugar whole food diet. It was given by Sullivan et al. In 1958. The dental status of children between 7 to 14 years of age residing in Hopwood House was included in the study for a 10-year follow-up period. And they all lived in already a strictly natural diet. They had occasional servings of egg yolk, and they had absence of meat, rigid restrictions of carbohydrates, and all these restrictions also included reduced sugar supplements. So the meal were supplemented with the vitamins, and occasionally the sweetening agent only given was honey. At the end of 10-year period, 13-year-old children had a mean DMFT of only 1.6. So the corresponding of general population is 10.7, which is very huge. And although 53% of children of Hopewood House study are caries free, only 0.4% of 13-year-old in common school public people are free from caries. So the children's oral hygiene was poor, calculus was uncommon, and gingivitis is prevalent in 75%. So even though Hopewood study said the strict regimen has reduced dental caries, they had other dental diseases and which gave a beneficial for this longitudinal study. Coming to the next study, Turku sugar study. It is compared the effect of sugar, fructose, sugar, sucrose and xylitol on caries development. So the major 3-component sugar involved was sucrose, fructose and xylitol is a natural supplement which has reduced form of sugar. And it conclusively shows xylitol karyostatic properties is very high. The study was carried out in Turku, which is in Finland, and the aim of the study is to compare the karyogenicity of the sucrose, fructose and xylitol. So the basis is that xylitol is a sweetening supplements which we already use in now in a common public. So method is in 2 years of this feeding study, 125 individuals, young adults were involved, which was about 27 years of old, and they all consumed sugar in the form of sucrose, fructose and xylitol. Sucrose 35 people and fructose 38 people, xylitol 52 people. About 100 items including the pastries, candies, chewing gums and the pickles, mustards and even the coughs mixtures were also made with these sugar forms. This finding shows that after 1 year, the sucrose and fructose had equal karyogenicity, whereas xylitol they did not find any caries. By the end of 2nd year, caries has continued to increase in sucrose group, but in fructose it has stopped, but it is maintaining. The caries has not increased, but in xylitol there is almost no caries. Therefore, fructose was less karyogenetic than the sucrose, and xylitol there was no karyogenic or even anti-karyogenic because it has some reversal of early caries lesions also. So this shows the potential benefit of the xylitol, and xylitol can stimulate the activity of antimicrobial enzyme. And the drawback of the study, the mean age is only 27 years for this high caries study, but 27 years is not actually an active caries lesion area. And subjects were also not randomized, and each group had different age groups and different peoples, and the subjects knew which group they were in. So this has a common bias, and foodstuffs other than those in which sugar consumption was known, they were also consumed by the subjects. So other forms of sugar have also been consumed, which has not been noted properly. And coming to the 4th study, the hereditary fructose intolerance. It's an observational study. These individuals, due to the dietary necessitates, it serves as a natural intervention group. It is caused by remarkable reduction in the level of hepatic fructose 1-phosphate aldanase. It splits the fructose 1-phosphate into 3-carbon fragment, and which is further metabolized. So which plays a vital role if you have taken a sugar in form of fructose. So the person who is affected with this rare metabolic disorder will have to avoid the fructose or sucrose, because these ingestion of this food causes symptoms like nausea, vomiting, malaise, tremor, excessive sweating and even coma. So this is also called as fructosemia. And Newborn in 1969, he found and tabulated the prevalence of dental caries in these hereditary fructose intolerance patients of 31 members, and he found surprisingly that dental caries prevalence were extremely low in hereditary fructose intolerance patients. So Tristan D. Cunha. So it was done in the remote island in South Atlantic inhabited by the people of European descendants. In the early 1960s, there was a temporary evacuation in this region because of a volcanic eruption. But later, after a few years, this island was rehabited because the volcanic eruption has settled down. After that, some modern industrialization has come up with established industries in the island, and processed food also became easily available. And the residents were given the dental examination on this island in 1932, 37 and 53, and followed up in 62 and 66. And results showed surprisingly that in 32 and 37, where there was no industrialization, pre-volcanic region had zero prevalence of dental caries in first permanent molars of 6 to 19 years old. But in the year of 62 and 66, after industrialization, the dental caries prevalence has increased from 50% to 80%. So which shows the industrialization and modern sugars had increased the dental caries prevalence. So the key takeaway and further direction from this diet studies and the link of this diet with the dental caries, the prioritization of sugar reduction is very important. Limiting both quantity and frequency of free sugar and added sugar is more critical dietary intervention for caries prevention. So not only the quantity plays a role, the frequency and the time of interval you take the sugar is also very important. And promote a nutritional balance. So as the studies and intervention says, not only the sugar plays a role, there is also nutrition which plays a reversal role in dental caries. We have to encourage diet rich in the essential vitamins and minerals, especially calcium phosphorus, which is the backbone of the dental teeth, and the fluoride to support tooth development and remineralization. We have to educate the patients broadly. Empower the patients with knowledge about dietary impacts on oral health, moving beyond basic hygiene to comprehensive lifestyle advice. Integrate the evidence into the practice. Utilize the finding from human animal studies and block pH research to inform personalized dietary counseling in the clinical setting. So in the clinical setup, always individualized care, prioritize in experimenting and implementing this all in animal studies, saying that block pH has to be increased and the diet has to be reduced with refined amount of sugar, and frequency has to be limited, and also it should be given during the meal and not between the meal. Advocate for public health initiative. Supportive policies that reduce the sugar availability and promote healthier food environment aligned with WHO recommendation is very essential, and always have an atmosphere with healthier food environment. So the epidemiological evidence overwhelmingly confirms the central role of diet in dental caries. As a dental professional, it is our responsibility to extend beyond the treatment to proactive prevention through the comprehensive dietary education and advocacy. So thank you for watching this class.